How is progress in paratransgenesis coming along? That's what I want to cover today in Science Spotlight 14, Progress in Paratransgenesis. I'm Dr. Mark Williamson. Let's get started. So, to introduce the topic of paratransgenesis, it's important to also talk about transgenesis. And nowhere better to start than a review. 2010 had this review by Transgenesis and Paratransgenesis to control insect-borne diseases, current status, and future challenges. So both of those, transgenesis and paratransgenesis, are genetic approaches to vector-borne disease control. So we need to unpack that a little bit more if you're not familiar. So vector-borne disease control are diseases that are spread by vectors. And in this context, vectors are some sort of arthropod that will have the pathogen in its system, usually its gut, etc. It'll bite, say, humans and transmit that disease, transmit that bacteria, that virus, that other pathogenic or parasitic organism. So that's what transgenesis and paratransgenesis are focused on, vector-borne diseases and controlling those. And they're alternatives to more traditional, maybe more low-tech approaches. Those would be things like insecticides to kill those vectors, vaccines to protect against, say, viruses as a subtype of those diseases, and then there's other methods like that. And so specifically transgenesis is genetically modifying, because it's a genetic method, genetically modifying the insect vector to reduce transmission or some other format. So in this case, there's a nice little infographic here. So you have, say, the egg of a fly, you insert DNA in some way to genetically transform that larva can grow up to adult, it can breed with wild type and hopefully pass that on to offspring as transgenic adults as well. So you can reduce the ability for this vector, this arthropod, this insect to reproduce, to carry the bacteria or other pathogen, to transmit it, etc. There's a variety of methods. So that's the baseline, transgenesis. Now, the more complicated, though actually in some ways simpler, but conceptually more complicated, is the method of paratransgenesis, where you don't genetically modify the vector, you actually modify a microbial symbiont of the vector to reduce things like transmission. So in this case, you're targeting something that's found in the insect, in the vector, not the vector itself. So it could be a virus or it could be bacteria. You do some sort of genetic transformation, whether it's plasmid or direct DNA, and you have a genetically modified symbiont. Then you can get that into the vector in various different ways. So you can have paratransgenic adults, so adults that carry these transformed bacteria, viruses, etc. They can pass them on to their offspring, pass them on to other adults. So that's what that looks like in a nutshell. So where does this come from? Where was the first work on this? It was actually done back in 1992, so it's been over 3 decades. The first recorded work is in Beard et al. In this transformation of an insect symbiont and expression of a foreign gene in Chagas disease's vector, Rhodnius Prolixus. So I was not able to find a non-paywall version of this article, but I was able to get the abstract, and kind of pulling from that, we can get the big picture. So the researchers, Beard et al., were looking at kissing bugs. These are triatom bugs. They're insects. They're vectors of Chagas disease, which is caused by a trypanosome protozoan. And so the goal of this approach was to modulate the competence of vectors to reduce transmission. And to do so, they actually were able to infect the kissing bugs with transformed symbionts, so these bacteria, that express an antibiotic protein. And they were successful in doing this in the lab. Now, the oldest paper that I could find the full copy was in 1999, and it was looking at tsetse flies. So tsetse flies, those are vectors of African sleeping sickness, which is also caused by a trypanosome protozoan, and this affects both humans and cattle. And so again, the goal was to modulate competence of vectors and reduce transmission. Now, in this case, they did some micro injection into hemolymph of fertile females to actually get the bacteria in there. They use recombinant as symbionts, some sort of symbiotic bacteria, and they had green fluorescent protein transmitted to progeny with high efficiency. So in this case, they didn't actually express a sort of antimicrobial or sort of anti, I guess, for prozoan, antiprozoan peptide or protein or something, but they had a further back proof of concept where they had these symbionts express green fluorescent proteins, so you could physically see if the tsetse fly, especially their progeny, actually had these transformed symbionts and that they retained. So this was actually a more proof of concept than the beard at all, but did show that you could get genetically transformed symbionts into tsetse flies and they would pass them on to their progeny. So with that in mind, let's zoom out again to a broad review of Paratransgenesis. There's a 2022 overview of Paratransgenesis as a strategy to control pathogen transmission by insect vectors. This will kind of go through the big picture, how this actually works, nuts and bolts, what can be targeted, etc. So there's some good tables, pictures we'll go into in more detail. But the problem that Paratransgenesis is trying to solve is that a more typical approach to vectors control is chemicals, so insecticides, but insects are increasingly resisting them. They're getting insecticide resistance. And so that's no longer working as well. So you can't really control the vectors, these insects, these flies, mosquitoes, etc., by spraying for them and killing them. And there's all sorts of vectors that cause disease for humans. Mosquitoes are the biggest one, but there's other flies, like we've already seen tsetse flies. There's hemipterans, like kissing bugs. There's ticks. Ticks are a big one. And there's other insects and arthropods. And it's not just for humans, it's for animals, cattle, other livestock, and even agricultural plants. And there's a lot of different pathogens, bacteria, viruses, protozoans, etc. And for viruses in particular, now, for some viruses, you can use vaccines, but there's actually few vaccines available for vector-borne viruses. So that's another problem. And so paratransgenesis is a potential solution. Now, one of the real nice set of benefits over, say, transgenesis is that it actually has a wider vector coverage. Because if you imagine, say, there's 3 different genera of mosquitoes that cause a lot of diseases. To have a transgenesis control, you need to have essentially transgenic mosquitoes for every single species. Whereas if you could find a gut symbiont, a symbiotic bacteria that's found broadly in mosquito guts, you could have transgenic microbes that could maybe use in all the species, so all in one go. Additional benefit, it's a lot easier to produce a bunch of microbes than it is a bunch of insects. And there's also a potential for both horizontal and vertical transmission. A lot of like the microbiome is passed from the mother to her progeny, so that's the vertical. But see, if you have the microbiome, it can be spread across other individuals, like adult mosquitoes, etc., while feeding, and so that would be a horizontal transmission of these microbes. And a lot of targets, humans, livestock, agricultural plants, as we've talked about. And so this image here goes through how you would do this. So examples, a fly, it has a bunch of gut bacteria, say. You can pull out the bacteria and you could select microorganisms that are amenable to this approach. A couple different ways you can genetically transform to create a transgenic bacterium. You can use genomic DNA, so directly edit the genome, or you could use plasmid DNA. In either case, your transformation will have these bacteria with a special gene that you want to be activated. This gene to do something to reduce transmission. So it could be on the plasma, it could be in the genome. In any case, you have a modified plasmid genome, you have transgenic bacteria. Then from there, you can grow that up and you can infect the fly back. Whether it's oral infection, etc. You could even inject it, but an easier way is to bait it in, to eat it. It becomes part of its microbiome. And then there, the transgenic bacteria can express your gene, whether it's more in a more traditional route, where essentially it's the DNA to RNA to protein. So a peptide is sort of a short part of a protein or what have you. And that peptide, that expression of that protein would be something that would help interrupt a parasite transmission. Another way you can do that is, instead of having sort of a gene expressing DNA to RNA to protein, it could actually express double-stranded RNA, which serves as interfering RNA. So it could actually silence the expression of a target gene from a vector or parasite. Either way, the goal would be to block parasite development. And so that's what paratransgenesis does. Now, there are some requirements to make this work in the whole system. First of all, you need to have a culturable, symbiotic, or commensal microbe that you can actually take out of the vector, grow it up, and modify it. These are typically bacteria, but we'll see other examples as well. And that microbe should be able to colonize all insect or arthropod instars. Ideally, this isn't strictly necessary, but makes it much more easily for it to spread through the population. The microbe should be non-pathogenic to humans and animals. Doesn't make any sense to have this transgenic bacteria to stop some sort of pathogen if it is also pathogenic. Defeats the purpose. So it needs to be benign. It also, the fitness of the microbe should not be compromised after genetic transformation, because if the microbe can't compete with the wild type microbes and stay in the population, it's not going to do its job. And then the effector molecule, that this transgenic microbe, it needs to be made properly and secreted properly. So that's the term that's used in paratransgenesis. The effector molecule is whatever this transgenic bacteria is going to produce to either sort of knock down the pathogen, reduce transmission of the pathogen, or otherwise sort of make it less likely that humans are going to be impacted by that pathogen. And so that needs to be made and secreted and get to the right place that it can affect what it needs to affect. And then you need a way to introduce and disperse that modified microbe. If you can't get it into the population of, say, mosquitoes in the wild, it's not going to be very effective. And finally, there need to be approval for use in the field. So this is all well and good if you can show this in the lab, but to actually have a real world impact, you actually be able to sort of legally, ethically deploy this in the field to actually reduce infections from this pathogen in real life. All right, so table 1 is nice on this review. It's a summary of important advances in approximate chronological order. We see, starting out, we see Rhodnius prolixus. That is those tritone bugs, those kissing bugs that carry Chagas disease. Then we start to see Anopheles. Those are mosquitoes. A lot of Anopheles and kissing bugs being the main model systems for this. And then finally, this figure here really shows some of the hurdles. Certainly, you need to be able to show this in the lab, but you also need to simulate this in the field to assess risk, and you need to be able to consider social and political factors. You need to have community support. You need to have approval, because really, at the end of the day, if you don't have all these in place, you're not going to be able to have arthropod that is vector-borne disease control if you can't actually deploy it. And so while some of the science things seem to be really moving on, the real sticking point is actually a further downstream to actually get this approved. And as far as I know, at this point, there are no approved uses of paratransgenesis. So this is all, at this point, still essentially conjecture or a proof of concept that hasn't had any real world impact yet. All right. So let's switch gears a little bit to look at some specific work in specific vectors. One of the big ones, of course, is mosquitoes. And so there's a review in 2015 about this for mosquito vector control. So Anopheles, Aedes, and Culex mosquitoes, those are three genera that transmit malaria, dengue, and lymphatic filariasis. So really big diseases. Culex also, I believe, transmit West Nile. So if you're more worried in specifically North America. And now transgenic procedures are promising in these mosquitoes, but they're technically difficult. They're self-limiting and only work with one species at a time. So that's why Paratransgenesis are really promising. They're simpler, can work with multiple species, and then can be modified. So the symbionts can be modified so their fitness is not reduced. Some potential targets that go beyond what we've already talked about. Now we've talked about commensal bacteria, but actually they bring up the idea of fungus as being a target. One of the nice things about fungus is I believe it can actually directly infect through the cuticle of insects, so it'd be easier to get into the population if you could successfully create paratransgenic fungus. So I think it's a little more difficult than bacteria. That's why bacteria is the main use in paratransgenesis studies so far. But they also mention others. You could imagine viruses. Viruses are another idea, and even like eukaryotes, like protozoans. Now, they bring up requirements of bacteria for this. They need to have a effector molecule. We've already talked about that. A mechanism to excrete it, and the bacteria, of course, have to survive. And one notable candidate they brought up in bacteria is the genus Wolbachia. However, there's still a lot of knowns, and risk management is really needed for real-world deployment. So again, in Paratransgenesis and broadly, mosquitoes specifically, the biggest sticking point is actually being able to deploy this in the real world, and that has not happened. Other vector work, very interesting 2016 article about symbiont-mediated RNA interference to insects. So if you remember one of the images we talked about, there's kind of two main things you could do. You could have a gene that essentially will express a peptide, so a protein, but you could also have it express double-stranded RNA, and that's what's called RNA interference. RNA interference methods are powerful. They can interfere with gene expression in the vector or the microbe, but it's really hard to deliver double-stranded RNA directly to things. But if you used symbiotic bacteria and insects, you could be able to have continuous, constitutive double-stranded RNA synthesis. The bacteria are growing, they're constantly expressing this double-stranded RNA. You could also have trauma-free delivery. You could feed these transformed bacteria. You didn't have to inject the insects with the double-stranded RNA. And so there's a lot of benefits. And so they, in this paper, demonstrated this approach, symbiont-mediated RNA interference with two insect species. We've already seen the first one, a tried-home bugs. So it's a tropical disease vector, so Chagas disease. And they also used a very different insect, a globally invasive agricultural pest, also, which is the Western flower thripe. So this should be an agricultural pest, but could also be a useful target. They used a couple different bacteria, a gram-positive actinobacterium for the triatome bug, and a gram-negative gamma proteobacterium for the Western flower thripe. And they were able to see this. It kind of worked out. And they expect that this symbiont-mediated RNA interference can work with other insects. Now, I think something I wanted to look at specifically, because I know a lot of vector-borne diseases are caused by ticks. I went looking for specifically tick paratransgenesis, and it was hard to find many articles, but I did find this one from 2023 called Frankenbacteriosis Targeting Interactions Between Pathogen and Symbiont to Control Infection in the Tick Vector. Not surprised I couldn't find it because they use this funky term. The system they used was Oxydes scapularis. This is a tick vector. They were looking at the pathogen Anaplasma phagocytophilium, which causes the disease anaplasmosis. And their symbiont was a bacteria called Sphingomonas. At least that was the genre. And so they have a picture of what they did here. Essentially, both of those bacteria, both the disease and the symbiont, or the pathogen and the symbiont, compete in the tick gut, and they describe Frankenbacteriosis, so inspired, unsurprisingly, by Frankenstein, and as genetically transforming commensal bacteria in ticks to mimic and compete with tick-borne pathogens. Essentially, Frankenbacteriosis is paratransgenesis. They just used a different name, which made it hard to find. And so, yeah, what they're able to do is some metaproteomics to sort of pull out the fact that there's a pathogenic one here and a commensal one here. Did some genetic transformation, so you could have the Franken bacteria or the paratransgenically transformed bacteria to have this antigen. They did some tests and they found that it would reduce infection in cells and would lower the risk of pathogen transmission. And this paper was helpful in laying out some of the conceptual groundwork. They cited a couple earlier works by Jose de la Fuente, but those works were non-experimental, but they kind of lay the groundwork of people looking at paratransgenesis-like things in ticks. But for the 2023, the Frankenbacteriosis article, this was experimental, and they did find that they could get the MSP4 protein expressed in these transformed Sphingomonas, increasing infection and fitness in ticks of this commensal bacteria, and then reducing infection of the pathogen in human cell culture. That was a big picture. And they had it all streamlined if you want to look more into it, of exactly all of what they did. They did natural feeding, capillary feeding, and did, yeah, a lot of fairly good work here. All right, that's not all when it comes to other insects or other vectors. Ticks are arthropods, but not insects. There's this 2012 paper called Paratransgenesis, an approach to improve colony help and molecular insights in honeybees. And so this is actually an example of what I ran to, what I would call positive paratransgenesis, where you use paratransgenesis to enhance a host organism's positive effects on human health and economic incest rather than sort of downplaying a negative effect. So honeybees, you can try to put transformed bacteria in there to boost their positive effects, whereas you would take a transformed bacteria, say, put it in a mosquito to tamp down its negative effects, mainly transferring a pathogen. And so the concept for this was they wanted to transform one lactobacillus species, L. Kuhnke, for use as a vector to promote health of honeybees in functional genetic research as a sort of proof of concept. They were able to transform it, and it would secrete a reporter protein in different growth media, and the bacteria were fed back and were still found after a day of feeding. So they sort of succeeded in doing that. And so that kind of lays a groundwork for doing this more in bees or other beneficial insect species. And so that was proof of concept. I went looking for trying to find an actual paper that did this more in practice, not just a reporter protein, but actual proteins that might improve honeybee health. So I found this one in 2020. I could only find the abstract, but from there, I found that they were able to engineer a bacterium, it's not Gracella alvi, to produce double-stranded RNA to interfere with both a deformed wing virus infection and also interfere with Varroa mites, which are parasites on honeybees. So those are some actual experiments, not just proof of concept of the overall idea. So there has been some progress on there at least. All right, so we talked a lot, went through a lot of things here. So I thought it'd be good to review the conceptual space and fill in some of the cracks of what paratransgenesis can do and what all the different ways you can do it. So first of all, it's the actual pathogen that you're interested in. You can target that. Of course, the pathogens are borne by vectors, so you can target the vector or think of specific vectors. You can think about what the target is, whether it's humans, other animals, etc. You think about the symbiote, what are you going to actually use to transform? And then finally, the method of which you're going to transform that symbiote. So for pathogens, there's a variety of different pathogens that you might look into. Viruses and bacteria are the top ones. They are a lot of the pathogen-borne disease. There's also protozoans. Protozoans here being the colloquial term for single-cell eukaryotes, so like Plasmodium, that causes malaria, etc. Nematodes can also be spread by vectors. Even what I would call macromolecules, like for example in ticks, there's alpha-gal syndrome, which is caused either by I think the an alpha-gal carbohydrate or a protein. Anyways, it's a strange thing where it's a disease caused by a tick, but the disease isn't a pathogen. It's something else. It's a macromolecule. It's an organic molecule. So you can maybe target something like that. And then you could roughly put into everything else into other. There really aren't any known vector-borne platyhelminths. That's flatworms. That's another major parasite, but that's transmitted in a different method. There are some fungal parasites, though not of human ones, that are vector-borne. There's some evidence of that in agriculture and plants. So, like for example, Dutch elm disease, I think might be transmitted from vector insects biting plants. So those are some things you could target. Now on the vector, there's all sorts of ones. We've covered a lot of them. Mosquitoes are the big one. Tridome bugs, tsetse flies, sand flies, midges, black flies, so all sorts of flies, deer flies. You can think of more uh, sort of common parasites that you might not think can also carry disease, like fleas and lice. Chiggers, those are, I believe, larval mites. They can transmit some disease. Thripes, more for the plants. So we also saw the Western flower thripes. Ticks, a big one, I think understudy one. Other plant-feeding insects and other arthropods in general. So a lot of vectors to choose from. The target, or sorry, the symbiont. See, bacteria is the biggest one. I'd say viruses with the next one. There has been some discussion in the literature, though I haven't really seen any major work on it. Would be to use something like archaeal symbionts. So archaea. Fungi has been suggested, though I haven't seen a lot of actual work done. And then some more grab baggy things of like even talking about using protozoans or maybe something else, though that's much more speculative. And then targets. Clearly, humans are the main target, vector-borne diseases, but you can think of diseases of livestock, diseases of agricultural plants, and you could go down the line of importance, like pets, other animals, other plants. So there might be more sort of downstream or more niche applications of paratransgenesis. And finally, the method. Certainly, there's all sorts of specific methods you could use for, say, specific transformation, like CRISPR-Cas9 has become really important for direct genome editing. You could do more of that, other methods like that, but I tried to split it down to what I saw as sort of the broad scale different methods. We've already seen that you could bring in your gene through either plasmid DNA or genomic DNA, so that's one setup. And then you could typically have that gene express either a peptide or that double-stranded RNA. And so I would say there's four major combinations of plasmid versus genomic DNA and peptide versus double-stranded RNA. So with that, that's the conceptual space. There's a lot to work in here. Then I thought it would be helpful to actually look through, well, what sort of diseases are important? What sort of vector-borne diseases could we even target? And so I went on the CDC and brought out all their vector-borne diseases I could find. And so starting out here, there's ones we've seen before. Chagas disease, those are tridome bugs. There's trypanosomiasis, so that's sleeping sickness. That's tsetse flies, so we've seen those before. There's a variety of one, and those are all Discobian eukaryotes, as well as leishmaniasis, that's a sandfly. There's some by midges, black flies, deer flies, even fleas, like plague and typhus. There's a variety of other typhuses and other bacterial diseases. by a variety of things from chiggers to body lice, and then ones that have multiple vectors. So those might be useful to look at. Of course, the big vector would be mosquitoes. So we have a bunch of diseases here with malaria topping the list here. That's a big one. There's filariasis. That's actually a nematode. And then there's dengue, probably the biggest virus. And then there's a whole bunch of different viruses from Zika to West Nile to things you probably never heard of, like Eastern equine and encephalitis virus. And then finally, something I think that is really understudied in paratransgenesis, though it might be more difficult, is ticks. Ticks carry a lot of different vectors from eukaryotes, so protozoans, or babesiosis to bacteria like anaplasmosis, viruses, various viruses, probably the most well-known being some of these fever ones, but Lyme disease probably the biggest pathogen, bacterial pathogen, but you even have something like alpha-gal syndrome, which is not pathogenic. I mean, there's no pathogen, it is a disease caused by either a carbohydrate or a protein. All right, so closing out here, I want to think about some frontiers for paratransgenesis, like where it could go next, what could really be studied that isn't as well studied. And so this first part here, sort of the things I came up with, brainstorm from reading the literature and sifting through and digesting it. So first of all, there needs to be real world trials and validation. I understand that's the hardest sticking point, but until that actually happens and can get off the ground, this is all conjecture. This isn't solving any vector-borne illnesses until you can actually trial it and reduce it in real life. I think another frontier is leaning on that honeybee work of looking at more ways to use positive paratransgenesis. So use transformed symbionts to boost positive qualities of insects like honeybees or others. And I think that would be an easier selling point for real world validation. I think non-human beneficial targets is a good frontier. So looking more at how can we use this to protect livestock or protect agricultural plants. That might be an easier sell as well. Looking at non-bacterial symbionts, certainly viruses, fungus, maybe even protozoans. Tick vectors, I think, is a big frontier. There's not been a lot of research in there, and ticks spread a lot of vectors, especially in North America. That might be a really beneficial study. And as well as maybe looking at zoological reservoirs. I know ticks, a lot of times, ticks, their primary blood feeding is not in humans, it's in like rodents. And so if you could sort of target those zoological reservoirs where paratransgenic approach, you could maybe really knock it down and be easier than having to deal with sort of species or pathogens that don't have zoological reservoirs. All right, and then with a little bit of stretching even further the frontiers, I asked some AI models of maybe what future work in paratransgenesis might look at, just to see if there's any good ideas out there. Grok gave me one of them, microbial and genetic stability. I thought that was interesting. So seeing how long sort of transform bacteria or other symbionts can keep up fitness, and whether that gene, that transformed gene, will stay in the population or not, or whether it becomes deactivated after a certain time. ChatGPT gave me several bangers. Gut microbiota targeting. There's increasing evidence that dysfunction in the gut microbiota can lead to, or at least make worse, symptoms of IBS. Might be linked to things like obesity and even diabetes. So be able to target the gut microbiota with these sort of methods. A target invasive species was another idea that I thought could be useful. And then finally, this idea of anti-pathogen smart microbes, where the microbes, they're transformed, can actually sense and respond to the pathogen. And so we're only, say, expressing the peptide when the pathogen is present, otherwise not doing so. So that was some interesting frontiers. That here are references to all the papers that I looked at here. And thanks for joining me. I found this progress in paratransgenesis, while a little sad that it hasn't reached any real world impact. There seems to be a lot of work that has promise. So definitely comment and in the chat, in the discussion of what do you think, where it should go, what you thought was interesting with Paratransgenesis, and I'd love to hear a comment about what topic I should cover next. Till next time, thanks for joining me.